any questions, you can record it and discuss with Professor on next Thursday. Now I will play video.
Can I ask when will we have our midterm test?
Midterm?
Yes.
30 minutes, I think.
I didn't hear it clearly. Can you say it again?
Can you ask your question again? Hello?
Okay, I can't hear you clearly. Can you say it again?
And could you repeat your question again?
I want to know when will our midterm test take place?
Midterm cap?
Yes. Yeah.
Your meaning is midterm exam or the other thing?
I mean that the midterm test of our course, like the mid course.
Midterm test.
Yes, the test.
Okay. I don't know. You should ask Professor Huang. Because
Okay.
I see the schedule. Maybe let me see the schedule. Wait a minute.
Okay.
Oh, the schedule do not show the midterm exam. Okay, I think next Thursday you can ask Professor Huang, and I will tell her.
Okay. Okay, thank you.
So we can play video now?
Yes.
Thank you.
My topic today is involved in the microbial biochemistry. Biochemistry is very broad area for understanding the basic metabolism and also correlated with growth and nutrition in microorganisms. In this lecture, I will focus on bacterial biochemistry. And the first part, I will like to introduce the growth. Again, also focus on bacterial growth. The typical cell division in bacteria is called binary fission. As I show in the figure here, the replication requires the extension of the cell wall and replication of the chromosome. And then because of the septum form between the two parts and then fuse. So two identical daughter cells produced, and both daughter cell are new and also cannot be differentiated which one is parent, which one is a daughter. Okay, so this is how bacterial divide. So this is a sexual life cycle because the two daughter cells are identical from the parent. Here I will show you the two types of gram standing cells. The left-hand side is gram-positive bacteria, and the right-hand side is gram-negative bacteria. So the EM showing the Bacillus subtilis, okay, and the right-hand side is the E. coli about their division. So normally in the textbook we use cartoon to relatively clear to demonstrate the events, but EM is actually our original evidence and proof. Okay, so you can compare those EM photos to the previous cartoon on the previous slide. As for the how we measure bacterial growth, okay, because the bacterial growth from 1 to 2, 2 to 4, so from 1 to 2, so it's one generation, okay. So the cell numbers is calculated as 2 to the numbers of generation as shown here. So with number of generation increase, you will get the cell numbers shown in the middle part of this table. Normally, because the number is too big and not easy to be plotted to show their faces, that we will talk about this later. So we use log 10 to express the cell number when we analyze and then plot the cell growth curve. So the conversion of the number of cells in a population into the log expression of the cell numbers here, okay, is the way we analyze the bacterial growth curves. So here show you the bacterial growth curves using the two numbers of the cell number. The original cell number is showing on your right-hand side with the y-axis. Another y-axis is showing the log numbers, okay. As you can see here is the original cell numbers because of the binary fission. The dashed line show the number in log. So the x-axis is the generation numbers. According to the function of generation numbers, the cell, if in an active, healthy, keep dividing situation, you will get a straight line from a semi-log expression. So what is the generation time? The definition of generation time for bacteria is the time required for the cell numbers to double, okay? So this is a very easy equation to show the generation time is actually the growth time divided by the number of generations. But you have to be pay attention to this number. The cell has to be in the keep dividing condition. Therefore, the generation time will be accurate. Okay. So the method of estimating the generation time, G, is normally we plot a growth curve like this with the generation, with the growth time for bacteria normally in hours for the x-axis. For the y, as I mentioned, that normally we use a log scale of the cell numbers. So this is a semi-log scale to express the cell growth. So what we did is we actually get the slope of this straight line. Okay, so it has to be in the straight line area to estimate the generation time. Okay, so as you can see here, the growth time is 6 hours. Okay, and the cell double. Okay, so when the cell double, and it takes 6 hours, so the generation time is 6 hours. Okay, this is how we did when we do the experiments. But I will describe how we get a cell number in my later lecture. So there's also one properties that I need to address is that generation times vary markedly with the species of the organism and also the environmental condition, meaning different species of microorganism have different generation time. The same microorganism has different generation time when the environmental condition varies. Okay, so I summarize the typical bacterial generation time for you here. Is I list two types of microorganism. The first group is from prokaryotes. The second type is from eukaryotes. In prokaryotes, I pick E. coli, Legionella pneumophila, and Mycobacterial tuberculosis as the three representative one to show you that the generation time for the individual is around minutes, hours, and days respectively. That means for different bacteria, actually the generation time varies quite different. For eukaryotes, I use yeast as an example. For example, Saccharomyces cerevisiae, the generation time is also in the hours at 13 degrees C. Okay, so the generation time can range from minutes to days. There's another index that normally we will use to analyze the bacterial growth. We call growth rate constant. Okay, the growth rate constant representing the numbers of generation that occurs per unit of time. Normally we use hour. So 1 hour, in 1 hour, how many generations for this bacteria under what condition? Okay. Again, I need to emphasize that the cell has to be in an exponentially growth condition. Then the K constant will be accurate. So I'd like to introduce and spend a little more time about the cell bacterial growth curve. Okay. Usually we analyze the single pure culture in a closed system called a batch culture, meaning we don't continuously feed the nutrients in this system and we don't take out the cell or products from this system. This is a closed system. What happens for bacteria in this system to grow? Okay, so as I mentioned, again, this is important. Usually we plot the logarithm of the cell numbers versus the incubation time. As shown here in this slide, so the bacterial growth showing many different phases. Okay, again, here is the cell growth time in hour, most of the time, okay. And this is the logarithm of the cell numbers. So when the bacterial cell was inoculated in a batch culture, okay, under particular media, it will go through a lag time, log period, stational, and death. Okay, so again, this is grown under control conditions in a pure culture. So I'd like to explain more about the phases of the cell growth. First, if the bacteria is inoculated from a nutritious medium into a less nutritious media, you will have a lag time, and the lag time varies depending on the medium transfer. Okay, that means that bacteria need some time to prepare, to fit in and express certain gene in order to grow, and will be shown in the increase of cell number. Okay, but if the cell prepare, then to fit in the different media, it doesn't have to be shown quickly about the increase of the cell number. We call this a lag time. Okay, but it doesn't mean that the cell doesn't do anything. Okay. Okay, the second condition is if the bacterial transfer from the medium are the same to the medium are the same from the previous one. Sometimes you will not see lag phase, meaning bacteria already ready to get into growth in the new medium, but the media are the same. Okay, so sometimes the lag time is also disappear. We don't see the lag phase. Okay, so it happens. Okay, so for the log phase, I already explained quite detail about it because of the binary fission. So during the log phase, meaning the cell keep dividing without any limitation. So why the cell keep growing? Some situation happens in the media. First, the cell might produce some of the toxin into the media, which will affect the growth. Or the respiration of the cell will produce more carbon dioxide, which make the media more acidic if this is aerobic bacteria. So while the pH drop to a degree, the cell will not grow so as effectively as the fresh media. Okay, and third situation is when the limitation of the media happens, the cell growth also gets decreased. So during this time, the cell will gradually get into a phase called stationary. During the stationary phase, it means that the growth of the bacteria and the death of the bacteria reach equivalent, reach a balanced situation. Therefore, you don't see the cell number change. But it doesn't mean that that cell stop to grow. Okay, just it's a dynamic situation for quite some times, depending on the bacterial species. So some species have very long stationary phase because they will keep, they're likely to keep the metabolic rate very low. So they will keep their life, but they don't metabolize actively. Therefore, they don't divide, but they are not dying. So this situation happens during the environment. For example, in the river or in ocean, when the nutrients is limited, cell actually just keep in a very low metabolic rate, and therefore it doesn't look increase in cell numbers at this condition. So some cell stay in a condition like low metabolic condition. So many things happens during the stationary phase that I will describe later. So if the cell death over the cell growth, we call death phase. So as you can see, the cell number decrease according to the time. Okay, so we call this a death phase. Okay, since this is a closed system, we don't take the cell out, we don't put in the nutrients. So this is just showing in a batch culture how cell will perform their growth condition using the available nutrients. Okay, so the time varies quite a lot for different species, as I keep emphasizing. And this is important for today's lecture, so I put the stars on your slide. So during the nutrient limitation, okay, or we say starvation, what happened to the bacteria? Okay, so this is called the stationary phase. So many things happens during the nutrient limitation for bacteria. There are many biochemical and physiological change that I will summarize in the following slides. The first change is, as I mentioned, that the bacteria will keep a low metabolic rate. Secondary, because the protein expression at the different phases are different. So under the carbon, nitrogen, phosphate starvation, some protein are induced and expressed. So the total cell protein composition, of course, will be changed. The third one is during the stationary phase, actually the bacteria become more resistant to some environmental stresses, okay, such as high temperature, okay, osmotic stress, and certain chemicals such as hydrogen peroxide, solvents, and pH. So the bacteria, when they starve, they know they have to be careful because they don't want to die. This is life, right? So the resistance to environmental stresses increase. I'm going to tell you more about the change, but before that, I'd like to remind you that bacteria is a single cell life. So it's obviously life want to keep alive, right? So during the nutrient limitation, they still want to keep their life. So this is how they adapt to this condition. In addition to the previous change, the cell size also decreased. Okay. It's because the bacterial cells, which enter stationary phase upon the starvation, generally become smaller. That means the bacteria will divide, but they don't grow bigger. Okay. For example, the bacteria in Russia will become cocoid shapes. This cell size change happened in many genus, such as Pseudomonas, Arthobacter, Klebsiella, Escherichia vibrio. Okay, this is also help the cell to get more nutrients because when the cell gets become cocoid, the service and volume ratio increase, which will help the cell to get more nutrients more effectively to the cell. So the change in surface properties of the bacteria also happens when the bacteria is starved. The cell surface become more hydrophobic. This is and are more adhesive. So the nutrient limited cell to adhere to particles that have increased the possibility to absorb nutrients on the surface. Okay, so what happened during the surface change become more hydrophobic. It's because the membrane phospholipids has different composition in fatty acids. Okay, for example, in E. coli, when in the starvation, the unsaturated fatty acids will be methylated in their double bonds to form cyclopropyl type of fatty acids. Okay, so the fatty acids composition was switched during the nutrient-limited conditions to become more hydrophobic. So far, the advantage of this switch is unknown. However, we can get some understanding from the mutants that cannot synthesize the cyclopropane fatty acids. These mutants appear not to survive on the stretches as well as the wild type, such as starvation, different oxygen tension. Therefore, as you can see that bacteria should prepare not to die during the nutrient limitation. And the nutrient limitation is actually it happens in the environment. Normally at lab, we provide excess nutrients because we want to grow the cell, do some experiments, and we want to get good cell numbers. But during the different cell phases, then you will actually have harvested different cells, even from the same species. This is very important when you conduct experiments. In summary, bacterial population follow a sequential series of growth phases, namely log, stationary, and days phases. The knowledge of the growth phases is critical to understanding for individual bacteria. Okay, in the population dynamics, then you will know that how the population goes up and down and what's the interaction between the bacteria with the environment, such as nutrients, and how therefore we can do the population control. And in the full reservation and the sporage, this is also important. You know how we can keep the safety of the foods. It's also important for industrial microbiology processes such as fermentation. For example, ethanol production, you have to know what's the status of the cell, because different cell phases actually have different efficiency to ferment or to produce the products in a larger scale growth condition. When we talk about the importance of having a growth curve for bacterial culture, as I mentioned, that it's important for the large production of some products such as antibiotics. So here we show you the with the consumption of sugar as a nutrients, the cell grows according to the growth phases, which including very little left and starting with the log phase, stationary phase, and the death phase. The green line show the example as antibiotics such as penicillin. The penicillin is increase only when the cell near the onset of stationary phase. Okay, so it's not correlate with the cell well. Okay, so for the antibiotics production, such as this as an example, if you don't monitor the growth phase well, then it's not easy to understanding the physiology about the biosynthesis of the antibiotics, as well as the base harvest time, and of course for the base profits. The secondary metabolites are defined as metabolites. They are produced near the onset of stationary phase and not produced by all the microorganisms. So for the primary metabolites, they are produced during the active cell growth phase. They match the growth in population of the organism well, such as this example. Primary metabolites are produced by all microorganisms. Okay, so this is an example why the growth phase is important for the production of biotechnology or pharma products. I like to explain a condition called biotic growth and also Catapult Repression in the presence of glucose. Normally, the students, they like to use glucose. They think glucose is the best carbon source in the experiments, particularly when we learn about chemistry for the catabolism of the macromolecules, such as polysaccharides, proteins, lipids, is our nutrient, right? Then the following degradation will have sugar, monosugar, amino acids, fatty acid, and glycerol, respectively. So in our textbook in biochemistry course, normally they only use glucose as a starting catabolism nutrients to explain how bacteria or how our cells, even in eukaryotes, to get energy from glucose. Therefore, in our mind, we think glucose is the best sugar for bacteria to grow. Okay, so this is true for certain bacteria, but not for all. As for the first phenomena influenced by the concentration of glucose, called biotic growth. In E. coli and many bacteria, they can prefer to grow on glucose first. So their genes involved in the catabolism of glucose are constitutively expressed in the cell, meaning if the presence of glucose in the media, the glucose will be used first. Okay, and this study is a very famous example in E. coli. Remember, this is in coli. In the presence of both lactose and glucose, E. coli will use glucose first till they use up glucose. And then, so this is the first growth phase of the E. coli is actually on glucose. So if the glucose concentration decrease, the cell reach the stationary phase. It's actually also the lag phase for another sugar such as lactose in the media. So the bacteria will grow on the lactose as a second phase. So it actually is a two growth phase using solely the preferred sugar such as glucose and then the lactose. Okay, so as I mentioned to you, the growth curve is from one batch culture, but in this batch culture, there's the presence of two types of sugar. Different bacteria will have different preference. We call this two phases of growth called diauxic growth. This is a example from the E. coli. Okay. So the second phenomenon I'd like to introduce, which is also important, we call catabolite repression. Normally, students, it's difficult to remember the term. This term is more accurate and popular to describe this situation by the glucose replace other sugar to be used. So it means glucose repression, but the term is normally called catabolite repression. So glucose replaces the synthesis of enzyme required to grow on certain alternative carbon source. It happens quite often. So, and it also inhibited the uptake of certain other sugars in the cells. So these two phenomena will actually explain how the glucose lactose situation, as I described previously. Okay. The reason is the glucose concentration and the catabolites follow up from glucose catabolism will actually infect the adenylate cyclase, the enzyme which catalyze from ATP to form cyclic AMP. So when the cyclic AMP concentration change, it can regulate the expression of the other sugar. So the catabolite from glucose catabolism actually repress the other sugars expression. So this is how catabolite repression term comes from. Okay, but I would like to draw you attention that some bacteria actually prefer to grow on organic acids than on the carbohydrates such as glucose. For example, Pseudomonas aeruginosa, which is an important pathogen, and also another very useful and well-known environmental species called Putida. They like to grow on organic acid than on glucose. So when you grow this type of bacteria, we don't use glucose as a sole carbon source. We actually provide it with other acids. This is because the Pseudomonas had different glycolysis pathway from the normal well-known E. coli. So different bacteria actually have different glycolysis pathway. Okay, so this you have to bear this in mind. So we know the growth phase.
Okay, we can take a rest for 10 minutes